My name is Dedhia. I am the Director of Sleep Surgery at the University of Pennsylvania in Philadelphia, Pennsylvania. I'm an Associate Professor of Otorhinolaryngology and Sleep Medicine. I've been practicing here for about 6 years, and the research that we're talking about relates to my interest in obstructive sleep apnea and pathophysiology. DISECAD stands for Drug-Induced Sleep Endoscopy, Characterization of Airway Dynamics. So I'll start with the first part. DISE, or Drug-Induced Sleep Endoscopy, is a diagnostic tool that we use clinically and now in research to help identify the causes of sleep apnea in patients that are looking at things like sleep apnea surgery. So it's an important diagnostic tool. It happens under propofol. So we give the propofol, make patients sleep, and then we look with the camera, and in this case, we added physiology measurements while they're in a sleep-like state. And so that CAD, the DISECAD, is characterizing aerodynamics, and that's a novel approach where we're looking at things like airflow and pressure, in addition to the video, to understand what's happening to the patient's physiology during the sleep-like state. That's DISECAD. The data set, therefore, includes different layers. In the DISE itself, we're collecting, in a sleep platform, we're collecting different signals, such as their airflow through their nose, airflow through their mouth, the amount of effort they generate, which is we have a catheter in the airway to measure how hard patients are pulling during that time. And then also we're measuring things like oximetry. So we have really a sophisticated sleep study during this propofol sleep, and those are the signals that are being exported to the NSRR. In addition to the sleep endoscopy, patients also have an overnight PSG. So we sent over data to NSRR looking at the patient's sleep study results, so we can make some comparisons between natural sleep and drug-induced sleep. And all these patients in the DISECAD data set were those that already had the Inspire implant placed. And the data set was to support the research question of what are some predictors from DISE, for example, that could tell us about who might respond to this Inspire device. The data set allows people to look at physiology during drug-induced sleep endoscopy, or DISE. And these are in patients with Inspire, but there are two parts of the DISE. There's a part where Inspire is not on and a part where Inspire comes on. So you can look at the baseline run, or the run with Inspire, or the run with CPAP. So you've got various interventions during a drug-induced sleep endoscopy with these very intensive monitoring to see how physiology changes over the course of time, propofol as well as with the interventions. The second application would be how do things during DISE relate to the outcomes from the overnight sleep study. That was a split night sleep study. The first half of the night was without therapy, then with therapy, so we can determine clinical response, and also can look at the physiology signals during the PSG and make comparisons back to DISE. We looked at different avenues, BioLink, NSRR, and it seemed like NSRR was the most appropriate. Dr. Redline is a colleague of mine and also aware of her involvement with it. So it seemed like a way to share data. In the end, really the goal here, it's not a zero-sum game. We want to try to improve and allow others to make insights that we didn't have the time or resources or knowledge to make in the data set. So giving that to other people is a really important part of science for us. And so this is a nice avenue to do that. I think data sharing, I'm learning from some of the feedback you guys have given us to our team about data cleaning. So it's not just dumping a data set. I mean, NSRR has sort of proven that it's more than that, we got to make sure that the data can be presented in a way that others can access it. So I think it's data sharing. It sounds easy, just dump data somewhere, but I think in the case of NSRR, which is the right thing to do, it has to be primed for others to analyze it. So I think there's a process there that takes time. This is a translational project, and what that means, I have a clinical practice of sleep apnea surgery, and to understand how to best select patients for that, this kind of data is really important to figure out. When we look at the signals that we collect in the DISE exam, how do those translate to outcomes in the patients? So population health, of course, sleep apnea is a big population health problem. Within that, people that can't tolerate CPAP is about half of our patients. We're talking about a very large number of patients that are struggling with CPAP, looking for other options, And the drug-induced sleep endoscopy exam with this physiology as part of the NIH grant can help us understand things that we can't see with our video endoscope. It's the signals that really make up the disease, and drawing parallels between natural sleep and drug-induced sleep. I think that is going to really help our ability to predict patients that are appropriate for different treatments.